better. So yes, you can already understand we did more or less the same thing in the pigs as we did in the chickens. So we also had a look on chicken gut function in relation to residual feed intake. Overall, there are so many physiological features that are already related to differences in residual feed intake. There's, from my perspective, a little bit more evidence for the chickens than for the pigs. And the digestive and absorptive processes just present a small part of these plethora of features that are involved. For the chickens, there is more evidence available for the duodenum as for the jejunum, and you will see that this is the reason why we also focused in the study more on the jejunum than on the other parts of the gut. Another thing that we wanted to clear in ECO-FCE, and you've already heard from the gut microbiota study that chicken experiments were conducted at 2 different locations. So we really wanted to understand what difference exists in gut functions or similarities are there among different contemporary populations, as most times only one rearing environment was used to select animals with extreme RFIs. So our hypothesis for the gut function was that chickens of equal RFI would have analogous differences in intestinal size, structure, and function when raised at different experimental sites under controlled conditions. Like for the pigs, our objective was to investigate the nutrient digestion, gastrointestinal size, jejunal permeability, and related gene expression in animals of divergent feed efficiency. The experimental design was already presented by Zina, the previous speaker. So we used common protocols, common genetics, common diets before we selected the animals at mid-uni and AFB. So, then we collected feces for digestibility from day 34 to 36 of life, and did the collection of intestinal tissue, liver, and pancreas from day 37 to day 42 of life. Regarding the lab analysis, we used the acid-insoluble ash as digestibility marker and determined the protein retention. And for the mid-uni chickens, we also determined the real protein in feces to differentiate between the protein originating from the digestive tract and from uric acid in feces. So here you can see again what kind of analysis were performed for the different tissues. So length and weight measurements as well as the histomorphological measurements were performed for all gut sites, but then we focused on the jejunum for the permeability experiment, again the disaccharidases activity measurements and candidate gene expression. The results were analyzed using an ANOVA and the mixed procedure in thus, and besides the RFI's fixed effect, we also looked up the location and the two-way interaction. However, not all data were, or all measurements were performed at both locations, and this is why we only have histomorphological results and permeability results for the mid-uni chickens. Regarding the results, we've just seen the detailed RFI values for AFB and mid-uni chickens. Here are only the average values again, and they came along with difference in total feed intake in females and males. Of course, with the high RFI chickens having higher feed intake than the low RFI chickens. We didn't see a difference in total body weight gain, but what we saw was a strong location effect for the total body weight gain, the chickens at weight mid-unit had gained about 300 to 400 grams more per day than the chickens at AFB. Regarding the protein retention, we saw trends for higher protein retention in the low RFI chickens, and of course they came along with a trend for lower excretion, because we could distinguish for the weight mid-unit chickens in the protein that came from the gut. This is shown by the weak protein excretion, and we could deduce the uric acid excretion. And so these data confirm that the difference among low and high RFI chickens was mainly on digestive tract level. However, when we combined the data from the weight mid-unit chickens with those from AFB, we again had a location effect. So AFB chickens had a 10 to 15% higher retention of protein, dry matter, ash, compared to those chickens raised in Austria. Regarding the histomorphological parameters that were only determined for the Austrian chickens, we could establish effects for the females. The males did not show differences. There you could see that low RFI was showed smaller villi and shorter crypts throughout the small intestine. The question again here is this just related to feed intake, or is this really a differences among the chickens of different RFI values? We also determined here the mucosal enzyme activities at the brush border in the jejunum. What you can see here is that we have a very strong location effect, but no RFI effect. What you can also distinguish is that we had transfer interaction and one interaction between RFI and location. And the significance was for the males, where we could see a difference from low to medium and high RFI for the mid-unit chickens, but no effect for the AFB chickens. And although it's just a trend, we had opposite results for the sucrase activities at the jejunal brush border. Regarding the results for the permeability, here again, like in the pigs, we could see that we had an increased paracellular permeability in low RFI chickens compared to high RFI animals. In addition to what we could establish for the pigs, where only the paracellular permeability markers clearly show differences. Here also the electrophysiological parameters, the short circuit current, which gives an indication for the ion flux, and the tissue conductance, which is also a permeability measure, showed similar trends for higher permeability and flux. Regarding the candidate gene expression, they were again in relation to the innate immune response, and because we saw good differences for the Washington chamber experiment, we had a closer look on the tight junction proteins and also determined the expression of a short chain fatty acid transporter. Regarding the tight junction protein expression, here again, like in the pigs, we couldn't distinguish differences for the IF groups at gene expression level, but we could see a decrease in the monocarboxylate transporter 1 but only for the male animals in the jejunum. When we looked at the innate immune response in the chickens, what became apparent was first that we had, again, these RFI and location effects, indicating that the effects were always stronger in the chickens from AFB compared to those at Remedioni. So, for example, in the females, we had a lower expression of interleukin-1 beta compared to with females with low RFI compared to females with high RFI. In males, there was then an effect for toll-like receptor 4, again, maybe indicating a different lipopolysaccharide abundance or response in the jejunum, which was higher with a low RFI compared to the high RFI animals. And also here the intestinal alkaline phosphatase responded, but this effect was, as I said, stronger for the AFP chickens compared to the Remedioni's chickens. But overall, these results give an indication that the gut microbiota seem to be involved. Just to sum this up, so we saw improved protein retention with low RFI. I haven't presented these data. We had to reduce liver weight, also with low RFI chickens. We saw for the females a response in relation to the structure of the small intestine. There was also this permeability effect, where the effect was stronger in the females and mostly absent in the males, and also the innate immune response, which might be related to the gut microbiota, was differently regulated. So to conclude, so our data showed that not only that there was a pronounced sex effect, but that the environment played a very important role regarding the environment, and we just discussed it in the discussion of the last talk. In general, low RFI was characterized with apparently reduced energy and nutrient needs for the small intestine, but enhanced nutrient absorption, apparently nutrient absorption in the jejunum. Overall, we could show that also there are differences in the jejunal structure and function, and with this could fill this gap of knowledge. As I said, the jejunal expression of our candidate genes indicated this impact of the local intestinal microbiota. So to conclude, to give an overall conclusion, our chickens had a similar RFI and a similar feed intake, but the underlying mechanisms may differ in relation to the environment in which the animals were reared. And with this, I would like to thank again the colleagues that were involved and you for your attention.